Welcome back to the third episode of Genetic News Now. Today we will be discussing point mutations. Point mutations result in the substitution of one base pair for another. The two main kinds of point mutations are transitions and transversions. A transition is a purine to purine or pyrimidine to pyrimidine, while a transversion is a purine to pyrimidine or a pyrimidine to purine. Today we will be looking at the given DNA sequence below. To better understand point mutations, we need to translate this DNA sequence to visualize how point mutations affect the translated amino acid sequence. Thus, here's the properly translated sequence. Now that we have the translation of the non-mutant strand, we can visualize the effects of a point mutation at the protein level. The first effect that we analyze is a missense. Missense is described as changes to the amino acid that may alter protein function or result in a non-functional protein. As shown in the sequence below, a transversion occurs at the A to T base pair, which was substituted for the G to C base pair. This results in the formation of an AGC codon, which codes for serine. Serine replaces asparagine in the protein chain. The next effect that we will look at is nonsense. Nonsense is described as a change to a codon that results in the codon becoming a stop codon. As shown in the sequence below, a transversion occurs as the G to C base pair was substituted for the A to T base pair. This resulted in the formation of a UAG codon, which is a stop codon, which is shown in the truncated sequence below. The next effect is called a silent mutation. A silent mutation codes for the same amino acid. In other words, this particular point mutation will not cause any changes to the protein sequence. As seen in the sequence below, a transversion occurs at the C to G base pair, which was substituted for a T to A base pair. Although the codon changes from AAC to AAU, each codon still codes for asparagine. The last effect is called a read-through. In a read-through, a stop codon is changed to a codon that codes for an amino acid, resulting in a longer protein chain. As seen in the sequence below, a transition occurs at the TA base pair, which was substituted for an A to T base pair. The UGA stop codon is changed to AGA, which codes for arginine, which allows the sequence to code past this point. I hope this shed a little more light on the subject of point mutations. Thanks for watching Genetic News Now.